And okay, for the last talk of this session, we're going to switch topics a little bit and talk about an aspect that's been a little bit neglected so far, which is the parasitic species that can live inside ciliates. And the talk will be given by Kassandra Zaila from the Hamilton College.
Thank you. I am Kassandra Zaila, and today I'm going to tell you a story about Rickettsia endosymbionts in Ciliates. Ciliates are well known to harbor prokaryotic microbes, and their relationships can be subdivided into three groups. One, symbionts can be as a nutritional source. Symbionts can be used as a defense, and symbionts can also cause environmental modifications. The best known example in this community is the killer symbionts found in Paramecium that provide the host with the ability to produce toxins that are capable of killing and outcompeting the symbiont-free competitors of the same or closely related species. The microbe we were interested in studying was Rickettsiae. Rickettsiae is a gram-negative bacteria that has an obligate intracellular lifestyle. Rickettsiae is well known as a causative agent of insect-borne diseases such as typhus, scrub typhus, and Rocky Mountain spotted fever in humans. However, recent studies have shown that non-pathogenic rickettsial species can be found in green algae as well as other ciliate species, as shown here by the in situ hybridization photos. On the top left, you have this little arrows that are pointing to the green. Those are the endosymbiotic Rickettsiae in the green algae. Then we have Ichthyophthirius multifiliis on the top right, and the pink represents the endosymbiotic Rickettsiae. We have, other examples in Diversities oligotryphs as well as Paramecium caudinum. This is a phylogenetic tree reconstruction using the 16 SSU sequences to show that the Rickettsiae-like bacteria forms a strong support sister clade to the pathogenic Rickettsia I spoke about earlier. This whole study focused on the species in the Rickettsia-like clade that is called the Canadactys Megaira, right here. What was particularly interesting about this clade was how dynamic it is compared to the other ones. As you can see here, Canadactys Megaira can be further classified into three subclades. The first subclade, Canadactys Megaira polyxenophilia, includes symbionts in both marine and freshwater ciliates, like Paramecium and Euplotus, as previously heard. And the other two subclades, B and C, can harbor a diverse host and habitats, such as ick, green algae, as well as be found in U.S. lakes, just to name a few. While it may seem that Canadactys Megaira is widely spread, it remains unclear how ubiquitously they can be found in environments, and their distribution of these bacteria within one species remains less studied. So the take-home message from this slide is that Canadactys Megaira can be subdivided into three subclades, and that they can be found in diverse hosts and habitats. So how about the distribution of Canadactys Megaira in one single host? Kaffoon and colleagues did some extensive surveys on Cattleya and Volvix cattleyi, and they found that the Canadactys Megaira may not be present in all isolates of one species, as it was only found in 12 strains of 4 unicellular green algae species and in only 1 of 9 strains of a multicellular green algae. The authors also suggest that the non-phagotrophic species did not actively acquire Canadactys Megaira, and these endosymbionts were inherited vertically. Once they got into the host, possibly through wounds or bites, despite these results, what becomes interesting is that Cattleya and Volvix cattleyi are non-phagotrophic green algae. And to our knowledge, no other research has studied the distribution of Canadactys Megaira in phagotrophic species, and therefore that is what we set out to do. We used the Ciliate Ichthyophthirius multifiliis, also known as Ick, as our model organism. Ick is the causative agent of the white spot disease that affects freshwater fish. In 2009, Sun, in Ted Clark's lab, detected endosymbiotic shingobacteria and Rickettsia Alphaproteobacteria in 2 strains of Ick from 2 isolates in the state of Georgia. The Rickettsia was later identified as belonging to Candactis Megaira subclade C, as seen here. Therefore, the questions to consider in this study were if Candactis megaira can be detected in most, if not all, strains of the phagotrophic ick. We used 18 strains that were collected from infected fish in the US, Taiwan, and Brazil. The strains were named with letters denoting the state or country of origin and a sequential number in which they were discovered. For example, Arkansas 1, if you see ARC 1, it means it's the first strain isolated from Arkansas, from the state of Arkansas. Those that you see here that are highlighted in red and underlined, they represent the strains that are derived from pet shop aquariums. Some of you that attended last year's conference in Italy may recall that my colleague Eliza McCall and others used mitochondrial sequences and successfully classified ick strains into 3 genetically distinct groups. Most Arkansas groups belong to the group 2, which is right here on the red. You can see, and then with the exception of ARC9, that is here in group 1. You can see how distantly related they are to each other. Arkansas strains are mostly derived from local hatcheries, and their evolution may reflect repetitive local infection, whereas the strains from New York and Taiwan mostly came from pet shops and appear to be represented in all 3 groups. Their dispersed evolutionary patterns suggest that they come from different sources. Endosymbiotic bacteria 16S rDNA sequences were either PCR amplified or derived from whole genome assemblies. 3 strains, G15, ARC11, and ARC12, were genomically sequenced using next-generation sequencing, and they were assembled by Megaira. All other strains were PCR amplified with rRNA primers, and that were specific for bacterial SSU rDNA or in-house designed primers specific against Rickettsiae. The PCR products were cloned and sequenced, and reconstruction phylogenies were done using maximum likelihood and Bayesian analyses. Here are our results. What you see here is the consensus tree of our newly obtained sequences. So you ask, can Rickettsiae endosymbionts Candactis megaira be detected in all 18 strains of ick? Yes, we did find at least 1 Candactis megaira in all 18 strains of ick. Was only 1 Candactis megaira species found in only ick strains? No, and I will show you a couple of examples. In 1 ick strain, we may find species, for instance, in ARC2 that's highlighted here in this zoomed in version of the phylogeny, is that we can find 2 sequences that are very closely related of the same isolate. Also, most ick strains could only detect Candactis megaira from 1 subclade. However, a few strains in particular, those derived from aquariums such as NY4, NY7, and Taiwan 5, were inhabited with symbionts from 2 subclades, most likely reflecting their complex travel histories and their exposure to different environments. Another question we considered was, how does Candactis megaira transmit? If transmission were solely vertical, we should expect congruent evolution between Candactis megaira and the ciliate host. In order to visualize these distributions of both, we map the groupings of ick strains to the Candactis megaira phylogeny using colored bars, as seen in the next slide. After mapping, we found some interesting results. For one, each Candactis megaira subclade could detect ick from all 3 groups, as shown by red, green, and blue bars in each subclade. Also, remember that Arkansas strains were part of the red group, with the exception of ARC9. We can see from here that Arkansas is inhabited with the Candactis megaira subclade C, with all of the other Arkansas samples. This suggests a strong local influence. Now, what did we learn from Candactis megaira? Well, we learned that with all of the new sequences, we now know that there are 4, not 3 subclades of Candactis megaira. 3 of these 4 subclades are capable of inhabiting ick, and subclade B is the only one that has not been detected in ick. Candactis megaira polyxenophilia was not known to inhabit ick. However, here we show ample examples of that. To date, rickettsial endosymbionts in subclade D have only been detected in ick. To summarize the evidence, the Canadactys megaira is ubiquitously present in 18 strains of ick. Canadactys megaira can be classified into 4 subclades, and based on comparisons of the phylogenies of both ick and Canadactys megaira, it is clear that the mode of transmission of Canadactys megaira is not solely vertical, as strains of ick in the same phylogroup may be inhabited with different subclades. The example of Arkansas strains of ick support the conclusion that local Canadactys megaira may transmit horizontally to ick newly introduced to the environment and outcompete with Canadactys megaira already inhabiting ick. For those strains derived from the aquariums that inhabited more than one subclade, it may be a reflection of the strain's complex travel histories and the aquarium and the exposures to different environments. Like many other research, our results actually raise more questions. They suggest that the association between Canadactys megaira and ick are more dynamic than previously ever thought. For some, we have several questions remaining. What role or roles does Canadactys megaira play in ick? What mechanisms account for the presumptive horizontal mode of transmission? Can Canadactys megaira subclade C or D inhabit ciliate species other than ick? What mechanisms determine the tropism of Canadactys megaira? This is a collaborative project with Indiana University, Hamilton College, Chai University, Martins from Brazil, and Academia Sinica, as well as Cornell University. On a side note, my professor Tang is forcing me to make this announcement that Hamilton College will be recruiting a 10-year track geneticist in the fall. Thank you, and I will take questions now.
So I guess I should ask a question. Am I right that this bacteria is, there are no strains that you didn't find this bacteria in?
Yes, it was ubiquitously present in ick.
Okay, so
In all strains.
and so that suggests that it might be symbiotic, in fact.
Yes.
Okay, and is there any? Do you know anything about what it may be doing for the parasite?
Well, we didn't really study the relationship, right? And that would be the next step. But because it's ubiquitously present, it could have maybe a dependent relationship, but it's something that we don't know at the moment.
And did you ever try curing it?
No. We
Curing
didn't.
ick of that to see what happens?
No, we didn't.
Okay, thanks.
I'll ask a question too.
Thank you.
So I'm just wondering, in your sequencing of this endosymbiont, if you maybe ever came across any bacteria or bacteriophage-like sequences in the endosymbiont. It's more of a curiosity, and I know it's not
Well, we
related
did
to
find
it.
other bacteria, but they were in low abundance, and they weren't ubiquitously present in all of them.
But no phage-type sequences in
No.
endosymbionts.
Okay, I was wondering if there's any evidence in the ick genome for an advantage of having these symbionts. Like, for example, if there are some essential genes that are missing in the ick genome, and maybe this function from these essential genes can be provided by the symbionts. I was wondering if you looked at that. If
No, we didn't look into that, but that is something really interesting to look into. At the moment, we just know that they're ubiquitously present. I guess that's kind of like the next question we can think about is, since they're so present in every ick strain, there must be a relationship here, right? So it may be a supportive relationship, it may not. So that's really interesting. Thank you.
Well, thank you, and thank you
Thank
to
you.
all the speakers of this session.
Thank you.